Welcome back to the deep dive. Ready to jump into a story that's kind of like a medical mystery. We're going to explore the beginnings of medical research, and we'll be focusing on a woman named Henrietta Lacks.
Henrietta Lacks's cells, they were taken without her consent, and actually they became the very first immortal human cell line.
Really?
Yeah, so imagine these cells could just keep dividing and replicating outside of the body. Huge for science, but how it all happened raises some pretty serious ethical questions. Things we'll definitely get into.
For sure. So our mission today is to really understand the whole context surrounding Henrietta Lacks and how her cells ended up making such an incredible impact on medical science. So where do we even begin with a story like this?
Well, let's go back to the early 1950s for a minute. Try to picture a time when doctors and scientists were really wrestling with understanding cervical cancer. There were a lot of big debates about how to diagnose it, how to treat it, the whole thing.
So what were the main sticking points in all these debates?
Okay, so one really key figure right in the middle of this whole debate was this guy named Dr. Richard Wesley Talind. He was like the leading expert on cervical cancer at the time. And he was a big believer in this idea that carcinoma in situ, which is a non-invasive form of cervical cancer, he believed that it was an early stage of the much more aggressive invasive carcinoma.
Okay, for those of us who aren't doctors, can you explain that a little more?
Sure. So think of invasive carcinoma like roots of a plant that have burrowed deep down into the soil. It's taken hold, it's growing. But carcinoma in situ is more like a seed that's just sitting there on the surface. It hasn't spread or invaded anything yet. But Dr. Talind, he was convinced that it was only a matter of time before it did become invasive.
So he thought it was best to treat them both the same way.
Exactly. He was all about aggressive treatment right from the get-go, including things like hysterectomies, even for cases of carcinoma in situ. His reasoning was, this is the only way to really make sure the cancer doesn't spread. But not everyone was on board with that.
I can imagine. So what are some of the arguments against Dr. Talind's approach?
Well, a lot of his colleagues felt like carcinoma in situ was often totally harmless and didn't require such drastic measures. They felt his approach was way too extreme, and it could actually cause more harm than good.
It sounds like a really tough position for women facing this diagnosis to be in. What were their options at the time?
Well, in 1941, this revolutionary screening test called the Pap smear came out. It was developed by this guy named George Papanicolaou, and it had the potential to find those precancerous cells before there were even any symptoms.
Oh, that's amazing. Sounds like it could have been the answer everyone was looking for.
It really did. Early detection meant doctors could step in early, maybe even prevent the disease from developing altogether, but there were some big obstacles in the way.
What kinds of obstacles stopped the Pap smear from becoming like a regular part of women's health care?
Well, first off, a lot of women just didn't have access to it. It wasn't something that doctors routinely offered, and healthcare wasn't readily available for everyone, especially for people in more marginalized communities.
Right, that makes sense. So even with a test that could potentially save lives, not everyone had equal access to it.
Exactly. And even when women did get a Pap smear, a lot of doctors didn't really know how to interpret the results correctly. They weren't trained well enough to tell the difference between precancerous cells and just normal variations.
So it was a combination of access issues and a lack of expertise that meant that the full potential of the Pap smear wasn't really realized.
Right. Meanwhile, Dr. Talind was super determined to prove his theory about carcinoma in situ. He wanted to collect more and more evidence to convince everyone he was right.
So how did he go about doing that?
Well, he launched this big study at Johns Hopkins where he reviewed tons of medical records and biopsies of women who had been diagnosed with invasive cervical cancer. He was trying to find a link between their current diagnosis and any signs of carcinoma in situ from earlier biopsies.
Sounds like he was really on a mission. Did he find what he was looking for?
His research did show that a really significant number of women with invasive cancer had a history of carcinoma in situ. But he wanted to go even further with his research.
What did he have in mind?
He thought that if he could actually grow both normal and cancerous cervical tissue in the lab, that would be the ultimate proof to back up his claims.
That's really ambitious. Wasn't it super challenging back then to grow living tissue outside of the body?
Yeah, it was. But luckily for Dr. Talind, there was another researcher at Johns Hopkins who was also trying to do something very similar. His name was Dr. George Gay.
Ah, Dr. Gay, the father of tissue culture. Wow.
That's the one. Dr. Gay and his wife, Margaret, they were pioneers in tissue culture research, and their big goal was to create the first immortal human cell line cells that could keep dividing forever and never die.
It sounds almost like science fiction.
I know, right? They were so passionate about their work, but they had been at it for years and years, and they still hadn't been able to do it.
So you've got Dr. Talind, who needs these living tissue samples, and Dr. Gay, who's desperately looking for human cells for his research. Sounds like a match made in heaven.
It really was. Their ambitions lined up perfectly. Dr. Tallinn saw a chance to maybe finally prove his theory, and Dr. Gay finally had access to the human cells he needed. But where does Henrietta Lacks fit into all of this?
That's what I'm dying to know. How did she become part of this whole story?
Well, Henrietta showed up at Johns Hopkins for her first cancer treatment on February 5th, 1951, and she had no idea that she was about to be part of something that would change medical history forever.
So what happened during that visit that was so pivotal?
So when she arrived, she was sent to the colored-only ward. It was a totally different time, obviously, and even at a prestigious hospital like Johns Hopkins, Black patients faced discrimination and segregation.
It's hard to believe that such blatant racism existed, especially in a place that was supposed to be dedicated to healing.
It really highlights how deeply ingrained these systemic issues were back then. They affected every aspect of life, healthcare included. Now, the standard treatment for Henrietta's cancer, which was invasive cervical cancer, the standard treatment was radium.
Radium. Isn't that incredibly dangerous?
It is. It's a radioactive element, and back then, people believed it had healing properties.
I know that exposure to radium can cause all sorts of serious problems. What were the side effects they were seeing back then?
Yeah, the initial excitement about radium kind of wore off as people started to realize just how harmful it could be. It could cause really bad burns, mutations, and even death in some cases.
That's terrifying. It's amazing to think that they actually use something so dangerous as a medical treatment.
It just goes to show how much our understanding of medicine has changed. But at the time, radium was this double-edged sword. It could kill cancer cells, but it came with these huge risks.
So how did they actually give Henrietta the radium treatment?
Okay, so picture this. They have these thin glass tubes filled with radium, and they put them inside these little pouches. And get this, these pouches were hand-sewn by a local woman.
Wow, that's a crazy image. Hand-sewn pouches holding radioactive material. It seems so primitive compared to what we have now.
Right. It really shows you how far we've come in medicine. But before they could start the radium treatment, a young resident named Dr. Lawrence Horton Jr. came into the operating room. He was working under Dr. Talind.
Okay, what was Dr. Wharton's role in all of this?
So remember how Dr. Talind wanted to collect those cervical tissue samples? Well, Dr. Wharton was told to take 2 small samples from Henrietta's cervix, one from the tumor and one from the healthy tissue nearby. And they didn't ask Henrietta for her permission or tell her what they were doing.
That's unbelievable. So that's the moment, right? The moment that Henrietta's cells became part of history.
It is. They just made a note about it in her medical chart, like it was no big deal. And then one of the residents took the samples to Dr. Gay's lab. And at first, they just treated it like any other sample. They didn't think anything special of it. They probably thought it would die just like all the others.
It's chilling to think how casually such a significant event happened. It goes against everything we know about ethical research today.
Absolutely. Things were very different back then. What happened next in Dr. Gay's lab with those seemingly ordinary samples? Well, that's a story for part 2 of our deep dive.
I can't wait. We'll be right back to continue this amazing journey through medical history. Welcome back to the Deep Dive. So last time, we were talking about how Henrietta Lacks' tissue samples had just made it to Dr. George Gay's lab over at Johns Hopkins, and they were about to become like this totally unexpected key to his whole research project on human cell cultures.
Right. Dr. Gay and his wife Margaret, they had spent years, trying to grow human cells outside the body. And their big dream, their goal, was to create this immortal cell line, cells that could just keep dividing and never die.
An immortal cell line. What would that have even meant for research back then?
Oh, it would have been revolutionary, completely changed everything. Imagine having a never-ending supply of human cells to study. Researchers could really dig into the secrets of human biology, how diseases work, testing new treatments, all without having to rely on animal models.
That makes sense. It would be a whole new world of possibilities. But had Dr. Gay had any luck at all growing human cells before Henrietta Lacks' samples came along?
Well, he had been trying so many different things for years and years, different techniques, different types of cells, you name it. But most of them, they would just die. And the few that did survive, they were so fragile and difficult to work with, it was a real struggle.
I can imagine. It must have been pretty discouraging to keep hitting those roadblocks.
It was, but Dr. Gay and his wife, They were incredibly persistent. They never gave up on their dream. They put everything they had into their research. They truly believed it had the power to change medicine forever.
That's some serious dedication.
Ah.
So, okay, back to Henrietta Lacks' samples. What happened when they got to the lab?
Honestly, at first, they didn't treat them any differently than any other samples that came through. Nobody had any idea that these cells were going to be any different.
So there wasn't like a big aha moment. They didn't immediately realize they had something special.
Not at all. The lab technicians, Mary Kubitschek and Margaret Gay herself, they were probably so used to being disappointed after all those years of working with cells that just wouldn't cooperate.
Yeah, I can only imagine. It must have been tough to face failure so many times.
It was, but their methods, they were so meticulous and they never wavered in their dedication. And you know what? That turned out to be super important.
Okay, so walk us through what they actually did with Henrietta's cells. How did they try to grow them?
So Mary, she prepared the samples very carefully. She put them in these test tubes with a special solution called growth medium, which was full of nutrients. And then those tubes went into this warm incubator, which is like a little chamber that's designed to mimic the conditions inside the human body.
Like they were trying to create a perfect little home for those cells to grow in.
Exactly. The incubator kept the temperature just right, the humidity, the whole atmosphere, everything perfect for those cells to grow and multiply.
So then they just waited to see what happened. What did they see as they were checking on Henrietta's cells?
Okay, so here's where it gets really interesting. Unlike all those other cell samples they had worked with before, Henrietta's cells started to multiply like crazy. I mean, they were doubling in number every single day.
Wow, that's unbelievable. So they weren't just surviving, they were actually thriving in the lab.
Oh yeah, they were doing better than anyone could have ever imagined. It must have been an incredible moment for Dr. Gay and his team. After all those years of trying so hard, they had finally done it. They had an immortal human cell line.
That's a huge turning point. They had struck gold, scientifically speaking.
They had. But we can't forget about the other side of this discovery. Henrietta Lacks, the woman whose cells made all of this possible, she had absolutely no idea any of this was happening.
You're right. We can't overlook the ethical implications of how they obtained and used her cells. We'll definitely talk more about that later. But for now, let's stick with the science. What made Henrietta's cells so unique? What made them so valuable for research?
Well, on top of their amazing ability to just keep growing and growing, Henrietta's cells, which they ended up calling HeLa cells, they had some other really interesting characteristics.
Like what? What made HeLa cells stand out from all the rest?
First of all, they were incredibly tough and adaptable. They could survive and grow in conditions that would have killed other types of cells.
So they were like the superheroes of the cell world.
Ha ha. Yeah, exactly. They could handle anything. And another thing, they were really susceptible to viral infections, which made them perfect for studying viruses and developing vaccines.
Oh, so that means they were a key part of the fight against infectious diseases.
Exactly. In fact, HeLa cells were crucial in developing the polio vaccine.
No way. So they played a role in getting rid of one of the scariest diseases out there.
They did. And that's just one example. HeLa cells have been used in so many studies, cancer research, genetics, drug development, you name it.
It's mind-blowing to think that these tiny cells, taken from one woman over 70 years ago, have had such a massive impact on human health and scientific progress.
It really is. They've become one of the most important tools in medical research, leading to countless discoveries that have made our lives better and helped us understand the world around us so much more.
Okay, so we've established that HeLa cells were a game changer for science. But what about Henrietta Lacks herself? What happened to her after those tissue samples were taken?
Sadly, Henrietta's cancer spread very quickly, and even though she had treatment, she died on October 4th, 1951. She was only 31 years old.
That's so sad. It's a reminder that even with all the amazing scientific discoveries, there was a real person at the heart of this story, a woman who lost her life to this terrible disease.
It's so important to remember that. Henrietta's story isn't just about scientific progress. It's also about human dignity and respecting people's rights, even when we're trying to make new discoveries in advanced medicine.
Absolutely. It raises some really big questions about who owns our cells, who benefits from them, and who gets to make decisions about how they're used.
Questions that we're still wrestling with today as science and technology keep moving forward.
Right. So Henrietta died in 1951, but her cells lived on. They were growing and multiplying in labs all over the world. What happened to them after her death?
Dr. Gay. He was convinced that these cells could completely transform medical research, and he wanted to share them with as many scientists as possible.
That's pretty incredible, considering how hard he worked to achieve this. He could have kept them all for himself, right?
He could have, but he didn't. He wanted to share them freely because he knew they could benefit everyone. He sent vials of HeLa cells to scientists all over the world in these little test tubes. And from there, they spread like wildfire, growing and multiplying in labs everywhere.
It's like they became these little scientific ambassadors, sharing Henrietta's legacy around the globe.
I like that. That's a great way to put it. And as the cells spread, so did Henrietta's story. The story of the woman whose cells made this whole scientific revolution possible.
But it took a long time for Henrietta's family to find out about all of this.
It did. It wasn't until the 1970s, over 20 years after Henrietta died, that her family learned about HeLa cells and their impact on the world.
Over 20 years. How did they finally find out?
So researchers who were studying HeLa cells, they wanted to know more about where they came from, so they reached out to Henrietta's family and asked them for blood samples to do some genetic testing. And that's how the family discovered the truth about Henrietta and her cells.
I can't imagine what that must have been like for them, to learn about all of this after all that time.
It was a lot for them to process. They were proud of Henrietta and what her cells had done for science, but they were also angry and hurt that her cells had been taken without her consent and that they had been kept in the dark for so long.
Yeah, it's totally understandable that they would feel betrayed and taken advantage of.
Absolutely. Their story really brings up the whole issue of who owns our cells, who profits from them, and who gets to decide how they're used.
Henrietta's family's experience really forced people to start talking about the commercialization of human biological materials.
It did. And it led to some really important changes in how medical research is conducted. Thankfully, we've come a long way since then in terms of protecting patients' rights and making sure research is done ethically.
So what are some of the changes that have been made to prevent things like this from happening again?
One big change is that we now have these things called institutional review boards, or IRBs. These boards review all research projects that involve human subjects to make sure they're ethical and protect the participants' rights and well-being.
So now there's a system in place to make sure that research is done the right way.
Right. IRBs look at the risks and benefits, make sure everyone involved gives their informed consent, and they make sure that vulnerable groups are protected.
That sounds like a huge step in the right direction.
It is. And these days, informed consent is considered absolutely essential for ethical medical research.
For our listeners who might not know, can you break down what informed consent means in this context?
Sure. Basically, researchers have to give potential participants all the information they need about the research, including any risks and benefits. And people have the right to say no or to stop participating in the study at any time.
No more taking cells without permission.
Exactly. Patients now have the right to make informed decisions about their medical care and whether or not they want to be involved in research.
That's a big win for patient rights.
Definitely. But it's also important to remember that ethics in medical research is always evolving. As science and technology move forward, we'll face new and more complex ethical challenges.
It's not just about following the rules. It's about creating a culture of respect, honesty, and making decisions together in medical research.
Absolutely. And Henrietta Lacks's story is a powerful reminder of why all of that is so important.
Welcome back to the deep dive. We've been talking about how HeLa cells, the ones that came from Henrietta Lacks's tissue, have totally revolutionized medical research, haven't they? They've been like a key part of so many incredible discoveries like the polio vaccine and tons of advancements in how we treat cancer. But it's not exactly a simple story, is it?
Nope, not at all. Remember, Henrietta's cells were taken without her knowing or giving permission, which brings up all these really big questions about patients' rights, how responsible scientists need to be, and even like who owns our biological material. These questions, they're still so tricky, even today.
Yeah, definitely. Let's dig a little deeper into these ethical dilemmas, shall we? Back in the 1950s, when all this went down, what was the attitude towards informed consent? Was it even a thing?
Well, informed consent, as we understand it today, it just wasn't standard practice back then. It was a different world, you know? Doctors often made decisions for their patients, thinking they knew best.
So people like Henrietta were basically kept in the dark about their treatment and how their tissues might be used for research.
Sadly, yeah, that was often the case. Especially in those big public wards, patients were seen as passive, like they were just receiving care, not actively involved in making decisions about their own health.
That's a stark contrast to how we approach healthcare today. Now it's all about putting the patient first, making sure they're informed, and giving consent to everything.
100%. We've made some progress in recognizing that patients have the right to make their own choices about their bodies and their medical treatment. It's a huge shift.
How did this whole lack of informed consent in Henrietta's case actually affect her family?
Well, for decades, they had absolutely no clue about HeLa cells and how they were being used. It wasn't until the 1970s, more than 20 years after Henrietta died, that they found out the truth.
20 years. That's unbelievable. How did they finally learn about it?
Some researchers who were working with HeLa cells, they wanted to trace the cells back to their source, figure out where they came from. So they contacted Henrietta's family to get some blood samples for genetic testing, and that's when the family found out about Henrietta's cells and this whole story.
I can't even imagine finding that out after all those years. It must have been so overwhelming.
It was. They went through a whole roller coaster of emotions. They were proud of Henrietta and what her cells had accomplished for science, but they were also furious and hurt that her cells were taken without her consent and that they had been kept in the dark for so long.
Of course, they had every right to feel betrayed and taken advantage of.
Absolutely. And their story really throws a spotlight on that huge question: who owns our cells, who profits from them, and who gets to decide how they're used?
It seems like the experience of Henrietta's family really sparked a conversation about the business side of human biological materials, how they can be bought and sold.
You're right, it did. It pushed for some really important changes in how medical research is done. And thankfully, we've come a long way in making sure patients' rights are protected and that research is done ethically.
What are some of the key changes that have been put in place to stop something like this from ever happening again?
Well, one of the biggest is the creation of these groups called institutional review boards, or IRBs. They basically review any research project that involves human subjects, just to make sure it's ethical and protects the rights and well-being of the people involved.
So it's like a safety net to ensure that research is conducted in a way that's morally sound.
Exactly. IRBs look at the risks, the potential benefits. They ensure that informed consent is obtained from every single participant, and they make sure that vulnerable groups are given extra protection.
That sounds like a critical step towards preventing any more ethical slip-ups.
It really is. And today, informed consent is like the bedrock of ethical medical research. It's non-negotiable.
Just for anyone listening who might not be familiar, could you explain what informed consent actually means in this context?
Sure thing. Basically, it means researchers have to be completely upfront with potential participants about the research. They need to lay it all out, what the research is about, any risks involved, what the potential benefits might be, everything. And people also have the absolute right to say no thanks or to stop participating at any point. It's their call.
So no more taking someone's cells without their explicit permission.
Exactly. Patients have a say now. They have the right to make informed choices about their medical care and whether or not they want to be involved in research. Their autonomy is respected.
It's incredible how far we've come in terms of protecting patients' rights.
It really is. But we can't get complacent. It's important to remember that ethics in medical research is a moving target. As science and technology continue to advance, we're going to face all sorts of new, complex ethical dilemmas. It's an ongoing process.
It's not just about checking off boxes and following the rules. It's about creating a culture where respect, transparency, and collaboration are at the heart of everything we do in medical research.
Couldn't have said it better myself. And Henrietta Lacks' story is a powerful reminder of why all of this is so crucial. We can never forget the human element, the individual lives that are intertwined with scientific advancements.
This has been an incredible deep dive, so insightful and thought-provoking. As we wrap up our exploration of Henrietta Lacks' legacy, what's the one thing you hope our listeners will take away from this?
I hope they remember that Henrietta's story isn't just about scientific progress. It's about human dignity, about taking responsibility for our actions, and about remembering that everyone deserves respect, especially when it comes to their health and their bodies. That's what truly matters.
Beautifully said. Henrietta Lacks' cells may have led to incredible advancements in science and medicine, but her story also serves as a warning. It reminds us to never lose sight of the human side of things, to remember that behind every scientific breakthrough, there are real people whose lives are affected, and their stories deserve to be heard and respected. Thank you for joining us on this journey into the complicated legacy of Henrietta Lacks. We encourage you to continue learning about her story, to think about the ethical issues we discussed, and to consider how we can all work together to create a future where medical research is ethical, just, and benefits everyone.
Until next time, stay curious, keep asking questions, and keep on diving deep.